Okay, so this presentation is about cycles, phase analysis, and synchronization in population, ecological populations. And so what are, if you know, like oscillating dynamics in population dynamics is one of the fundamental modes. Like there were studies and they looked in all kinds of time series, and basically in 1/3 of ecological time series they found some kind of fluctuations, oscillations, so it's a dominant mode. But there's many different mechanisms and explanations which can explain why we have these oscillations here. I mean, you have trophic interactions, age structure, parasites, and so on. You can have external influences. And so in my research, I occasionally studied systems which have these population cycles. And I thought maybe it's a good idea to show here 4 case studies from my own personal work. So I'm not going to review all these possible explanations. It's not, I mean, there's a huge bunch of literature. I don't touch this. This is a very personal thing. I just show 4 case studies from my own personal research and try to show you the approaches that we did, how we try to understand and try to show you the way we think about the systems. Sometimes it's not a good idea to have many different things in one presentation here. So it's an experiment. It's the first time I give this presentation, and I hope it works. So it might be that because like I have to be short in each one of these, I hope I manage in time, but it could be very interesting to have these 4 different systems which could not be more different maybe in some sense, and see what are the common things and what are the differences. So it could work to have this one after the other. So what systems we have? I mean, we have 2 large-scale field data. One is the hare-lynx cycle in Canada. We have the cycle of Antarctic krill in the Southern Ocean. And we have 2 chemostat system, laboratory system. One with single cells, the other in a predator-prey system in a chemostat. And in some sense, they're all superlatives. I mean, we have here the largest predator-prey cycle in absolute time. Here in the experiment, we have the largest predator-prey cycle in number of periods. In the Antarctic krill, we have the largest predator-prey, or the largest cycle in absolute biomass. And in the single-cell phytoplankton, it's special because actually there shouldn't be cycles at all. And maybe it's one of the most cleanest measurement of a biological cycle. But we will see this. And one common thing in all this analysis is I try we always use, in this case is here, a phase analysis. So if we have some oscillating dynamics here, then we try to separate the dynamics into the amplitude and phase and try to extract the phase evolution of these systems. Okay, but we will see how this goes. So in the first case study, that's the hare-lynx cycle, the most famous classical predator-prey cycle in all ecology. And I started this in the group of Louis Stone when I was postdoc. So actually that was my first project in ecology. That's when I started ecology. We said, okay, let's look at the hare-lynx cycle. So we got access to data of the lynx only. And you see, like in time scale, that's here over 100 years. So it's really the longest recorded time series. You see here different colors show you lynx abundance, so to say, in different regions in Canada. So different colors here are populations which are hundreds or 1,000 kilometers separate. And where do we have this data from? I mean, that's the recorded number of pelts which were sold. So it's an economic index, which is a proxy for the abundance of the animals. And if you so want, this trappers there in Canada, they made a very long field experiment for us over 100 years, and this extreme weather in Canada, which we can use for ecological analysis. Okay, so and then the first observation, what you can see here is that the cycle is extremely periodic in phase. And I will quantify this later a little bit. So it's really, you have always the same period length, about 9 to 10 years. Yeah? Whereas the amplitudes are very erratic. The amplitudes are vary a lot. So you see, if we have fluctuations in an ecological cycle, these fluctuations can be differently in the amplitudes and in the phase evolution. So phase evolution here is very constant, whereas the amplitudes are irregular. And so the first thing then we wanted to analyze is we wanted to have a model which can reproduce this. And so we wanted to have a chaotic model, so we needed three trophic levels. We needed 3 state variables like vegetation and hare and the lynx. And this model we didn't try at this time, we were still very naive, it was my first project in ecology, didn't try to really fit this model to the data. I just wanted to have a model which kind of captures the fundamental mechanism of the system and reproduces like that we have this constant phase evolution but chaotic amplitudes. And basically this model here, I mean, it's a simple trophic interaction, we don't even have here a carrying capacity in the vegetation. And so this the mechanism of this chaotic attractor is similar to the Rössler system, if you know it. So here so it was known from the literature that the motor of the dynamics, that's actually the vegetation hare subsystem. This makes kind of predator-prey cycle. That's the motor of the system, and this gives the 10 years. And this is built in here as a kind of Lotka-Volterra model with here some functional, type 2 functional response. And then this will give an unstable spiral in UV phase plane. So in UV and vegetation here, you have an unstable spiral. And then you have the lynx system, and then we have this W star term. This means that the lynx makes a nonlinear control. So when the amplitude here gets large, then lynx numbers shoot up. And then the system, this cycle here is stamped, and the system is reset in the smaller amplitude, and so you get this chaotic attractor. Yeah? And this, I mean, so it resembles like this constant phase evolution, erratic amplitudes. And yeah, okay, yeah, you see attractor reconstruction from the lynx data. It resembles the system. The next observation you can see here is this synchronization. I mean, so you have these different regions in Canada, hundreds of kilometers separated, and they're really synchronized in phase. But the amplitudes are not synchronized. The amplitudes are only weakly correlated between these different regimes. And and now the question is, what can drive the system? Like, how is it possible that the system can be synchronized in phase so well, but amplitudes are irregular? So one common explanation is it should be external forcing. There should be some external fluctuating factor in the environment with 10 years time scale, and this should synchronize all these populations. And so the only kind of known time series we have is the sunspot numbers, and the idea, it's actually from the literature, that I didn't come up with this, so the idea is that sunspots, like we are speaking about populations in the very north, where where the solar flux maybe could influence vegetation, and so it has also a time series of 10 years. And then what people did, they correlated this time series here with the time series of the animals. But if you correlate to time series, you integrate amplitudes and phases. But what we have seen, like amplitudes and phase have very orthogonal roles. So what we tried is to extract the phase evolution out of this system. And so we did a very simple thing. Basically, phase evolution, you just count number of oscillations. And we can count this because we have cycles where you have pronounced peaks. So you just detect the local maxima in the time series. And then you say always from one local maxima to the next, we have done one cycle. We have phase evolved by 2 pi, interpolate by a straight line in between, and it doesn't matter really how you interpolate, because for the frequency phase evolution, you just need to mean the slope of this line over many cycles. So you can really, in this very simple way, you can extract the phase evolution of such a cycle if you have only one dominant frequency in the data. And of course, if you have noisy data, you need to smooth, because otherwise you don't know what is your peak. I mean, so there are some details in the analysis, but it's a very simple approach. And then what you see, like when we go in we subtract here, that's here for the 7 different lynx time series, the phase evolution. And I subtracted already the mean phase, so we are in a co-rotating frame, so where basically they have the same, basically their frequency is 0. And so you see all these different links and in the different areas, they have basically absolutely same frequency, they're perfectly synchronized, whereas the sunspot has a different frequency, phase evolution is different. So the sunspot cannot be the force of this. And so what else, if it's not an external force, or at least not the sun cycle driving them, what could be the case? I mean, it could be local coupling. Yeah? And so we did patch models where we here, in this case, only 2 populations. Later, we were interested to couple many more, but I show you only when you couple 2 populations. You couple them by dispersal of the animals, of course, only are here. And then the interesting thing is like here that you couple them, and if coupling strength is 0, so they're totally independent. Yeah. And then even if you start in the same initial condition, because like parameters differ a little bit, so after some time, these populations, of course, the cycles go out of phase here. And if coupling strength is large enough, then both systems do exactly the same, and they're correlated in phase and in amplitudes. But interesting, like when you start to increase the coupling, the first thing you observe is that these 2 systems, they synchronize only in phase. So there's always a constant phase difference between the 2 populations, whereas the amplitudes can be still uncorrelated. And this is the phenomenon of chaotic phase synchronization that was just at this time, it was discovered in other chaotic systems. And we showed that this also can explain synchronization in ecological data. So that was very early study, like it's more than 20 years ago, so like when I entered ecology. And, but I think that was kind of the first analysis or first study where phase analysis was used to attack ecological time series. So that's just the first case study. So then I came to Germany, I got a research grant, and I got my own research group at University of Potsdam. And in this, then because it was such a big success with this synchronization, and what we wanted to do in the lab by, actually here somehow some things here, it's not like in my computer, so Ursula Gaetke, I'm not sure if you can see it, she had a lab in the biology, and she was willing to run experiments. So I got the funding for the experiment. I had the money, but she had the lab. And she was willing to do experiments with me. And actually, I wanted to realize predator-prey experiments in the laboratory and couple them in weird geometries. Yeah? So I was still optimistic that we can do all this. And so but I will come to the predator-prey system later, but it turned out we put this in the chemostat, we inoculated this, and it was just a mess here. So we looked at this, oh Jesus, I mean, it's no predator-prey. And then we tried to understand the system better, maybe to tune the system, control it better. And we decided just look at what just algae were doing. So we took single species phytoplankton, monoclonal phytoplankton in the system and study what they're doing. So to see, can we at least control what the phytoplankton is doing? And so in the chemostat, it's the most simple system how you can put an ecosystem in the lab, You have your mixed culture, you have an inflow of a limiting medium, in this case it's nitrogen limited, and then you have an outflow so that you have a constant volume. And so what could you expect? Let's first look a bit at theory. I mean, the theory for chemostat is well established. I mean, you have equation for your nutrient, equation for your phytoplankton. You put in here your functional response of choice, and you can run it. And this equation gives you the nitrogen either free in the solution or the nitrogen fixed in the populations. And that means like the total nitrogen in the system, either free floating or in the populations, after some transients, the total nitrogen should correspond to the nitrogen in the external medium. So after a certain transient, we should have a conservation of our nitrogen. And this means we can get rid of one state variable, and then you have only one equation left for the phytoplankton here. And this one equation, it can only asymptotically go to an equilibrium. So this system cannot do oscillations or something. We just have the only dynamic mode which is left, go asymptotically to an equilibrium. Yeah? That's why it's called a chemostat. It's a static chemical environment here. And that's also what we observed. You know, we made measurements. We had a particle counter, and so we could have had a sampling rate of 1 or 2 measurements per day. And then so you measure it, and then you run it for a week, and you see the sigmoidal approach to capacity. And actually, this dotted line, it's a fit of this growth model, so this fits perfectly. Yeah? And so typically here you would say that's it, yeah? And you're finished. But actually, I want to convince you there is a lot more going on here, and that's very often in science. If you don't measure something, you don't observe it. You wouldn't even expect that something could be going on here. And so the advance you do very often, even so I'm a theorist, but the advance you do if you have a new thing how to look at the system. And you know, we wanted to look at this because we knew our predator-prey system was doing this crazy stuff. So we developed here an automated measurement of light extinction. So we measured the turbidity of the fluid. So there was a syringe, we're controlled by a motor. We could take some volume out of the system and had a light beam and could look for turbidity. So totally non-invasive system. It was automated every 5 minutes. We could measure the turbidity, so kind of a continuous measurement. And this is how it looked here. So you have again a sigmoidal approach to equilibrium with a lot of fluctuations here. I mean, then, and if you look at this, you could say, maybe these are cycles, but you have to be very careful because we as humans, we always want to see patterns and data. And at this point, now you need to be lucky. Yeah? So for example, there's this kind of theorist like me, you better don't enter a lab, because when I'm entering a lab, you get data like this. But then we were lucky, we had a student, it was Thomas Massi, and he had the touch for the thing. And so he was working with this, and then after some time, the results looked like this. And from this point, you cannot deny it anymore, we have oscillations in the system. And the thing is now by the model, I explained to you we cannot have the cycles. So it means something in the model assumption must be wrong. We need some other state variable in the system. And what could it be? So it could be like cell quota, for example, like cells first load the nutrients. But this doesn't it doesn't lead to cycles. We could have viruses in the system. We could have host parasite cycles. It could be fast evolution, maybe something, or we could have different kinds of algae, some sticking to the wall, some floating, or whatever. But after some time, we believe that the key thing is here we have a synchronization. So again, synchronization. Now you see kind of parallel to the first thing, but of cell life cycles. And why would this be? I mean, when you have a cell and it wants to divide, so phytoplankton wants to divide. You can imagine this like the cell has to go a loop in some abstract space, and when it has done the loop, then you have suddenly from one cell you have 2 daughter cells, and there's several phases, and in particular this G1 phase, the first phase. There it's the phase where you have the new material for the new daughter cells have to be synthesized, and this G1 phase, that's nutrient dependent. And if you don't, nitrogen dependent, and if you don't have the nitrogen in the system, and this was our limiting resource, the cells cannot pass through this because it cannot synthesize the material it needs for replication. And now if you have a system and the nitrogen is depleted, then this kind of the cells there, they oscillate always in the cycle, they're stuck here in the G1 phase. And in this way, if you have now 100,000 or so cells in your system, they all get stuck in the same position, and then that's how you can synchronize them. And the first test of this is we treated the start of the system different. So before we inoculate the system, either we can give them unlimited resources, or before we starve them in their resources. And if before inoculation they're starved, we believe they're all stuck in this G1 phase. And then we put them in the medium, and they all start from the same position, and they all replicate in the same phase. And then we see oscillations. And if they were not starved, they were not arrested in their G1 phase, and then we don't see cycles. So yeah, so that was a very good first test. Okay, maybe this theory is true with the life cycles. But then maybe you all know this, when you try to discover something in ecology, it turns out somebody did this before you, at least in the models. There's always some guy who did the model before you. And so it turned out here, I mean, at least prominent people, we had Pascal and Caswell, they had predicted this kind of thing before. They had a paper, they really studied this in detail, and they predicted in such a system you should see these cycles. And they also suggested a nice experiment. They said, okay, start in a system which is where the cells are not synchronized, and then you make your perturbation, you switch off the dilution flow, the dilution in the system. And what should happen if you switch off the dilution? You also don't have outflows. The cells get not washed out of the system anymore, so cell number should increase. But at the same time, you don't get nutrients in the system, so the cells should get arrested in the G1 phase, and then we switch on flow, and then the system should cycle again. So that's what they predicted in their paper. So we did this experiment. So we did here two systems, one control and one other. And in the system which was mostly damped, so the red one was more strongly damped, we switched off dilution rate, the system cell numbers go up because they're not washed out. We switch on the dilution rate, and you see we initiated the cycles. Not so strong, it's a very small amplitude effect, but clearly we switched on the cycles. And then Thomas Massi, he experimented with different algae systems. So this was here Monoraphidium, then he did Chlamydomonas, it was already better. And then with Chlorella, I mean, that was amazing here. So that's unfiltered data, that's just a measurement of the system. And look at this, I mean the precision of the cleanness of the cycle for population dynamics, that's insane, I would say. It's unbelievable how clean you have the cycle here. When we increased the concentration in the external medium, we even could generate here sustained oscillations which didn't damp out, whereas typically, yeah, oscillations damped out here. So, and the amplitude is very small. But then it got even better, because look here, like what wonderful sine phase, that's measured population numbers. Because then we remembered, oh yeah, but we had our old measurement where we take the particle counter, where we can do measurements only so and so often, but there we get the size distribution of the algae cells. So we get a histogram of the sizes. And you know, like when in the cell cycle, the algae, they start very small, they get larger, 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 and then they're split in 4 daughter cells, and they're small again. And if you look at the size distribution, you see at these different points in time, you see like really a cohort of cells gets larger, 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 where some other cells still stuck in this G1 phase, and you see like this movement of this cohort. So that's the way the oscillation goes here. And then we could put this together. So here you see the experiment. Here your system goes to here capacity. We make our perturbation. We see our cycle, which is initiated. And here we see over time the cell diameter, so the cell size. So you see something weird is going on here, which I don't know in the beginning of the system. But then we get here stationary size distribution. Then we do our perturbation, and the cells get arrested in this G1 phase. So they're very small. And then but they're all synchronized in this very small thing. And then we switch off the dilution rate, and then we see here this cohorts, like cells, they're synchronized, all the cells increase in size, and we have the cycle. Yeah? So very nice experiment. And then we wanted to model this, and basically that was also done in this Pascal Caswell paper. If we just followed it up here. Like if in general, so like we want now to model the size distribution of cells. And I mean, I'd say in the ecological literature, it's established. I mean, you write down a partial differential equation for the size distribution. I mean, like you have the growth, this function g, which tells you how fast the cells grow in size. There you have a wash out, like that's why with larger size you have less algae, because like they're washed out of the system. And the reproduction is done by boundary conditions. So we say the size distribution of 2 pi, like when they have one rotation, is nu times of this is their size at phase 0. Yeah, so like the reproduction is just integrated by the boundary condition. But now the interesting thing is how does this growth rate, how does it work? So, and this is their picture from this Pascal-Caswell paper. So they say we have here one interval in this cycle, which is nutrient limited. So like here we have like a functional 2 nutrient limitation. And in the rest of the system, we have just a constant phase progression. And you can run the model. Here you see here you see the order parameter. So basically the kind of mean synchronization of all the cells. I know like the experimental data is in red and the simulation data is in blue, and you see here the cell numbers or the order parameter. And we kind of reproduce here what's measured in the system, only that here in the simulation we don't increase so much in cell numbers when we switch off the dilution rate. And then we can also look for the progression in the cell sizes and the cell size distribution. Again, here in this experiment, this typical way this cell cycle evolves in time, and we look at in the simulation, this reproduces very nicely what we see in the experiment. And now we can also make this kind of plot, and we see we very nicely can explain what's happening here. And actually, this case study is now case studies 2. That's all I want to say. But there is one. Let me just make a small excursion, like in synchronization theory, because we were speaking about synchronization before in the hare-lynx cycle. Like when you want to treat now this kind of thing like as a synchronization problem, we don't like in the hare-lynx. We don't have just 2 populations which we synchronize. We have many cells. So we synchronize individuals. But we have many. And in the synchronization theory, that's kind of the Kuramoto theory. You have the cell phase cycles, many of them, and they're all-to-all coupled. Every phase oscillator is coupled to every other. And then if you can now look for in this say that's in the complex plane, like your cells rotate here. You can measure the center of gravity, the order parameter, which is a measure of synchronization and the whole thing. Like if they're all in the same position, then you have here center of gravity is somewhere not at this origin, so you have some, the order parameter. And then you can rewrite the system that you don't have coupling all-to-all. That's just that actually every phase is just coupled to the order parameter, the phase of the order parameter. So it's more simple to write down the system. And then there's a wonderful theory going back to Kuramoto and Strogatz and other people, that you also write down the phase density of all the oscillators, and you have again a partial differential equation. But here the boundary condition is that that it should be continuous, the phase evolution. And so that's classical theory, and that's one beautiful theory to analyze this. And here I was thinking, like what we have here, it's a kind of flavor of this Kuramoto theory with some small differences. So first, like we have here a non-conserved number of oscillators. So typically in synchronization theory, you have always a fixed number of oscillators and you let them synchronize. But here the number of oscillators is not fixed, it's growing in time. So this means this is integrated by this different boundary condition. And then of course we need a loss term, so to conserve in total the number of oscillators. And then we have this, we don't couple to the complex order parameter, but we couple to the nutrients in the system. But one could do this a bit more simple if we just also say we just couple to the total population numbers. Maybe I put this here together. So on the left, that's the pure Kuramoto theory. very much studied systems. There's hundreds of papers study the synchronization of this. And actually what we have here is a slight modification of the system. It looks very similar. You know, we have a different boundary condition. We have this loss rate. The coupling here, it's a coupling to the complex order parameter, which has a phase and the amplitude. Here the coupling would just be to a scalar. And I try to solve this, and analytically I cannot. So if somebody here is very good with partial differential equations, I think that's something like one could really try to get a theory of this, because these equations write themselves down on its own. Like that can only be the most simple model. You cannot not make it more simple, and it just waits to be analyzed and with increasing coupling strength to see like the transition to synchronization. So if somebody here in the crowd has some experience and wants to work on this, or has a student who's able to, I would be happy. Okay, but that was, I hope that it doesn't crash. So if you solve this simplified system, and you see here, now it works, you see similar evolution of this phase distribution, very similar to what we saw in the other model. Okay, so that was the end of this second case study. The thing here is it's unbelievably precise. And it's also interesting because we have a synchronization here in the individual life cycles, but it has effect on the population level. So like it's the effect goes to a higher level of organization in the system, and maybe it could cascade even to higher levels. But I mean, like the amplitude is very small. Why should we care? And now I want to show you, I want to make a case now the same mechanism can give rise to very large amplitude systems. And we go now to the biggest oscillation we have on the planet in biomass, and that's Antarctic krill. So we had a project where we studied Antarctic krill, and you know, it's a key species in the Southern Ocean ecosystem. It's the most abundant animal species in biomass. Like if you have the years with a lot of krill, the whole Southern Ocean is red with this krill, with the krill swarms. I mean, but compared to the single species phytoplankton, it's an incredibly complicated system. We are in the Southern Ocean, so like we have this strong seasonality. Half of the year it's dark. We have ice in winter. The question is, how do this, the food of this krill is phytoplankton. In winter, you don't have phytoplankton there in the water is clean. But under the ice, you have phytoplankton. So the krill, they switch their food between summer and winter. And then you have different life cycles. You have juvenile krill, adult krill, and they have very different life strategies and use the food differently. So it's a really complicated system. And when you look at the data, you know, you see that's on a log scale. You see this pronounced fluctuations. So that's the largest population cycle in biomass, and the fluctuations are more than 2 orders of magnitudes. So I want to make, it was a study with Bettina Meyer, Alex Riabo from my group who did the heavy lifting in this thing, and Andre de Roos helped us to develop this model. And and why should this be related to what we had before? That's a study where people looked at the size distribution of krill in different years. And when you remember the size distribution we had before in the phytoplankton, it looks very similar. So maybe we have here a similar mechanism driving the system. And so we looked at this, and there's one long-term station in the west Antarctic Peninsula, the Palma series, and there you can download numbers of krill abundance and sizes of krill. And Alex was doing this, and we make, again, over some years here, where we got the data, we look for the lengths, and you see again here this cohorts increasing in size, very similar to what we had before in this algae system. Again, we have a similar resource starvation like we had in the algae system. So you see it in the recruitment index. So like we have here like young krill, and they get older. So krill is an animal that can get several years old. But in every year, these adults they produce eggs, and then you have juveniles. But these juveniles don't have food because the food is taken up by the adults, so they just die over the winter. It's not like in our phytoplankton system where the cells can just stay forever in the G1 phase. Here in the Southern Ocean, the juveniles, if they don't have enough food, they don't survive the winter and go away. And so we have this dominant cohort here, which totally takes or doesn't allow juvenile krill to develop. And only when this cohort kind of goes to small densities, like eventually because they die, then there's enough food and a new cohort can start. So that's basically the mechanism of the thing. And then we again took like a model where we try to get the size distribution, age distribution of the system, kind of very similar. I don't want to go in all the details, like maybe just show you so we have again this PDE. Now we have age and weight, so age and size, because this is not really coupled in the krill, because sometimes in winter even they get older, but the size gets smaller because they live in winter from their fat reserves. So we need to, you have here two state variables, age and weight. The boundary conditions is of course much more complicated, and you have different food resources, sea ice and phytoplankton, also ice algae and phytoplankton in the water. This is all seasonal, so it's a complicated model, but we can still solve it. And if you run it in the parameter ranges which you expect to be, and the model kind of reproduces kind of what we see in the data. So, so just to show you, then once you found some mechanism in one system, even if it's a very small amplitude thing in the laboratory, there's a good chance in ecology that you find an empirical system where you have the same mechanism running in some different flavor, of course, and even here giving rise to large amplitude cycles. Okay, so let's jump directly to case study 4. Stop this. Case study 4. We're going back to my experiments in Potsdam. You remember we were running this predator-prey system. We looked at this, it was doing crazy stuff, and we said we need to look at the phytoplankton more carefully. This was so exciting, the phytoplankton story. We're just looking at phytoplankton. I totally forgot about the predator-prey system. And then the assistant one day came to me, Bernd, what should I do with this predator-prey thing? I mean, we get bacteria in there. I said, predator-prey? What kind of predator-prey system? And yeah, we were running this for 1 year. I mean, okay, I mean, the biologists, they knew, but I, as a theorist, I totally forgot this and said, yeah, let's plot the data. So, so let's first, so we had algae and we had rotifers, that was the predator. We had this nitrogen as limiting resource. And then for this 1 year, they measured once per day, they measured algae and rotifer. They measured also the eggs, we had the egg ratio, and they measured the dead animals. And so let's plot it. And then we had this wonderful time series, and actually turned out over this 1 year, we had this predator-prey cycles going on. And it turned out we had a world record, because yeah, nobody was so crazy to measure a predator-prey system for so long. And the thing is, actually, when you look in literature, very often in experimental systems, people could realize predator-prey cycles, but only for a certain time, after maybe 10 or so oscillation systems were stamped. And even if you look in ecological, in the wild, in the field, we don't know how long a system can sustain this, because nobody could measure so long. In the hare-lynx cycle, you would many centuries to measure so many cycles. So we have a fundamental notion in ecology about predator-prey, but there was no empirical evidence that you can sustain in the order of 50 cycles. So in this sense, we had here directly a world record, but you see, it's a lot of fluctuations going on here. Yeah, so it's not clean. And it's not like in the hare-lynx, where we have this very clean synchronization or evolution in phase, and the fluctuations were only in amplitude. No, here the fluctuations are in amplitude and in phase. Here, so sometimes you have very period lengths of 3 days. Yeah? Typical period length is 1 week, and sometimes we have here maybe 10 or 11 days. So somehow the system is also very flexible in the period length. And to give you an idea, so here I separated the dynamics in a 90-day window where you can really clearly see what's happening. So, for example, here we have a phase of 4 cycles, and that's a textbook predator-prey system. I mean, perfect. I mean, of course, there is some trend. I mean, it's still a living system, yeah, but you have wonderful, you know, this 1 quarter period delay of the predator after the prey, you know, and one textbook thing. And then suddenly they freak out. Yes, so suddenly, I don't know what happens. Like, I mean, even though the prey goes down in number, the predators still increase. Yeah, so they don't care and just increase in numbers. And you know, you have this very small. I mean, like by eye, you could say, yeah, but isn't this like here? We have here some small peak and here the next. But that's kind of maybe wishful thinking. If you try to quantify this in such thing, you don't see predator-prey. But then they do it 30 days, and then they remember what they're supposed to do, and go back to something where you say, oh, clear predator-prey thing. And so we then distinguish. We say we have this coherent oscillations, and we have this non-coherent regimes, and it's not really clear what's going on there. And now, in order to analyze, we again try to analyze like with a phase approach, but we cannot do it so simple, like just detecting maxima. That's too noisy for this. And so we used wavelet analysis. So I don't know if all of you know about wavelet analysis. So here we have our time series, and we have here localized oscillating function. It's and try to see how does this time series can be fitted by a localized oscillating function. It's a little bit like in powers. If you make normal power spectra, you have a sine function of a certain frequency and try to see how good does this sine function fit your data, and then you get the power spectrum. But here we can do it localized, so we can move this localized function around in time, and we can change the period length, which is called scale parameter in wavelet language. And this is a complex function, so we have a real and imaginary part, so we also get information about the phase. Okay, and that's well established. I mean, Grenfell, 2001, he introduced this to study epidemiological data. It was studied in Ecology. There is a nice review by Cassel in 2008. So we used this approach. We did the wavelet analysis for here for the algae and for the predators, and but it's a complex number. You need to, if you want to visualize it, so you visualize the absolute value. So you just take the absolute value of the wavelet, and here you need also to average the thing a little bit, otherwise you don't see anything. And general remark, if you do wavelet analysis, you have to be very careful, because like we have here one-dimensional time series, and we make a two-dimensional plot. So this means there must be a lot of redundancy in this plot here. So this means a lot of the colored blobs you see here are just artifacts because you extract a one-dimensional thing in 2 dimensions, and one has to be very careful with statistics. But basically what we see over this time, like here, that's the period length from 2 days, so very fast, to 16, like 2 weeks. And we see typically here in a certain band, we have the most power. So typically we have an oscillation in the order of 7 days, but it fluctuates. So like there's no well-defined frequency. And if you average everything, you get the power spectrum. So in typical, we have a fluctuation of 7 days. But this is separately for predator and prey. Now you can look for the cross wavelet. If you take the wavelet, multiply the wavelet for the predator with the wavelet of the prey and average. And this tells you, do predator and prey fluctuate at a certain time scale, like with a certain frequency at some point in time. And you see, yeah, mostly they fluctuate together, but there's also areas where they don't fluctuate together. But the problem with the wavelet cross spectrum is this also depends on the amplitude of oscillation. So for example, here we have a strong amplitude in the predator-prey cycle, and this means we have also here a higher cross spectrum. It's like when you make the covariance function, also the covariance increases with, in order to get rid of this, like when you go from the covariance to cross correlation, you normalize by standard deviation. And so here you go to the wavelet coherence, you take this cross spectrum, but you normalize by the wavelets of power spectrum of predator and prey. So it's a normalized quantity. It's now it's also a real number. It's between 0 and 1. And this is really normalized and tells you if predator and prey cycle together or not. And then you see that's here this coherency. And here we see really regions where we have coherent oscillations, predator and prey really cycle together. And this was this one weird regime I showed you before in the Zoom where they don't cycle together, and the wavelet coherency directly tells you, no, here they don't cycle together. Then you have a regime again where cycle together and they don't cycle together. So like the wavelet coherency really picks us these regimes where they're nicely behaved. The thing is, but we wanted to have the phase difference between the two. But wavelet coherency is just a real number, so it doesn't give us phase information. But here, before we had the wavelet cross spectrum, this is not normalized, but it's a complex number. So from this, we can get phase information. So now the thing what we do is, so we go here in the wavelet coherency, and that's this black line here. We always look at which frequency do we have the strongest coherence. So that's this line. And then we take this certain frequency, go in this here, and from this we can extract the phase difference between them. So because like the frequency of oscillation is not constant in time, so we have always to look at what frequency we want to take the phase difference. And you shouldn't take it in these regimes because we don't have phase difference. And so we can now get the phase difference between predator and prey, of course only in the regions where they cycle coherently. And now we can take the histogram of this. And we do this in a circular diagram. So the green line here, that's the base, that's the algae. And now we take this red, that's the histogram here in the circular plot of the phase difference of the predators. And it's 90 degrees, so the predators come 90 degrees later than the prey with some variance. And from this analysis, we have only the predator, only the red thing here. But we have also the other life stages. We have here the eggs, we have the egg ratio, the dead animals, this is how they look. And so we can also make the same analysis for the other variables. And then we have here the circular histogram. Always, what's the phase relation of different stages in the cycle to the algae? So, and then you see the cycle starts with the algae. Then you get eggs, a little bit later, 45 degrees, 90 degrees later you have the predators, and a little bit later you have the dead animals. So it all makes sense. The weird thing is like the egg ratio. So the eggs per animals, they came before, and that was a very surprise. Why should the egg ratio come in time before the food? But you see it also in the data, like you see the egg ratio goes up, then the algae come, then the eggs and the predator. So it's really what you also see in the data. And so we believe that if you have such a cyclic community, such a histogram could be a fingerprint for the succession. So you can start it, analyze this thing also in other cyclic communities. And okay, then we did a model, constructed a model, and this model has now flavors of different case studies I showed you before. We needed an age-structured model because we have measured here eggs and so on. So we needed, in order to model this, we needed to have information of the different stages. So the model, we have the nutrients in the system, we have the phytoplankton, and here in the animal we have eggs, juveniles, adults, and dead animals. And then you can write down basically standard equations. And with the model, we tried to do so many things in the same time. We wanted to to have here a model which captures the thing, which processes which happening there. We wanted that all the abundances are in the correct order of magnitude. We wanted that the amplitudes are in the 7th day in the order of magnitude. We wanted to have this whole phase histograms that they are correct. We wanted to have that the egg ratio really comes before the other things, which meant we really had to take here the time delays, like egg needs to mature, and the time for juveniles to go to adults, that we needed to put in time delay differential equations. And I wanted to reproduce that the system occasionally switches from this coherent regimes to very weird uncoherent regimes. So we had tried to do so many different things and try to find a parameterization in the model structure which can get all of this. And this took me years actually to get done. But in the end, we got the model, so that's with our noise-free system, so the model reproduces what we see in the data. We get the circular histograms, which look also like in the real data. The tricky thing, I couldn't really find a dynamic mechanism for this going out of phase. So in the end, I throw so much noise into the data in the model that just by fluctuations, just by noise, suddenly you couldn't see the phase relation anymore. But I couldn't find a mechanistic explanation why it would have been nice if there would have been some intrinsic system, like from maybe bistability or something, where the system would switch to a different oscillation mode, which we couldn't get. Okay, and that's it. So I've shown you that there's 4 different case studies that you can have the cycles in systems, but it can appear in very different flavors. And maybe the biggest message here I have, when you study the system, an ecological system, you should always try to see what methods do you use. I know some people, they have one tool, and they always take this tool and apply it to a data. So they have a hammer, and every problem is a nail, and I don't care. I always go with the same. And I would propose, if you have some data you want to analyze, you have to see what questions does the data ask you, and then try to find the tools to address the data. And that's what we tried here in our work. Okay, thanks.